Alpha melanocyte stimulating hormone induced anorexia in Japanese quail likely involves the ventromedial hypothalamus and paraventricular nucleus of the hypothalamus. Caitlin McCann, Bryson Childress, and Nikki Creston.
Alpha-melanocyte stimulating hormone is an agonist to its receptor, melanocortin receptor 4, or MC4R. Stimulation of the receptor is central to the control of food intake. When alpha MSH is injected into the ventricles, a suppression of food and water intake is observed. These factors relate to animals that have previously been tested on, including rodents, chickens, and doves. The experimenters wanted to see if the same results would be observed in a more wild-type subject. In this case, the Japanese quail was chosen. The researchers were looking for effects of alpha MSH on food and water consumption, CFOS immunoreactivity, and gene expression, including factor mRNA. Background of alpha MSH and appetite. Prior to this experiment, it was documented that alpha melanocyte stimulating hormone played a large role in regulation of appetite. Tridos et al. Wrote in 1998 that alpha melanocyte stimulating hormone and others, which are expressed in distinct hypothalamic nuclei, have been demonstrated to suppress food intake after ICV administration in rodents. Alpha MSH also plays an important role in the regulation of food intake of neonatal chicks, per Kawakami et al. This earlier article that recognized the implications of alpha MSH on appetite was published in 2000. Since then, more has come to light about the effects of alpha MSH on appetite regulation. Alpha MSH has been shown to repress food intake in rodents, chicks, and doves. Background of alpha MSH and CFOS. McMinn et al. Found out that alpha MSH injection led to an increase in CFOS immunoreactivity in rodents. This was prevalent in the paraventricular nucleus. Klein et al. And Kawamaki et al. Found the same result in chickens. CFOS immunoreactivity was also found to be greater in the arcuate nucleus, dorsomedial nucleus, and lateral hypothalamus. Background of alpha MSH and mRNA abundance. mRNA amount has previously been found to be less after alpha MSH increase. This could be due to transcriptional changes in the nuclei. In the broiler chick study, mRNA was not affected at the melanocortin receptor 4. Since alpha MSH is an agonist, the presence of it reduces the abundance of mRNA. One specific example of this was shown by Starr et al., in which alpha MSH inhibited the production of nitric oxide synthase 2 protein. When the mRNA abundance was measured, it was found to have decreased.
Japanese quails as the subject. The researchers wanted to find out if alpha MSH had similar effects on a more wild-type subject that had undergone less artificial selection for traits, such as growth in chickens. The quails used in the experiment were caged individually after 4 days of life. The constants for the living conditions were a temperature of 35 plus or minus 1 degree Celsius, with 50 plus or minus 5 degrees humidity. The lights were kept on for 14 hours and off for the remaining 10 hours. A preliminary experiment was conducted to see if stress by using the restraint for affected corticosterone. It was found to not affect that. The quails used in experiment 1 were injected intracerebroventrically into the lateral ventricle with either 0, 0.55, or 50 pmol of alpha MSH. Food and water intake were measured every 30 minutes for 180 total minutes. For experiment 2, quails received either a vehicle or 50 pmol of alpha MSH. They were not given food post-injection. A cryostat was used to section the brains, and they were stained by immunohistochemistry using rabbit. For the third experiment, subjects were given either a vehicle or 50 pmol of alpha MSH. The brains were cut and RNA purified. PCR was performed. Results of alpha MSH on food and water intake. Quail injected with any amount of alpha MSH ate and drank less than the vehicle-injected quail at 30 minutes. The quail injected with 50 pmol of MSH ate and drank less than the control the entirety of the post-injection measurement. As you can see, the graph on the left shows food intake, while the graph on the right shows the water intake amounts. Results of alpha MSH on CFOS immunoreactivity. As compared to the control group quail that were only injected with the vehicle, the quail injected with the 50 pmol of alpha MSH showed about 3 times as many CFOS immunoreactive cells in the paraventricular nucleus and the ventromedial nucleus of the hypothalamus. There was no CFOS difference found between the experimental groups in the lateral hypothalamus, acute nucleus, and the DMN, as you can look at the graph. Results of alpha MSH on mRNA abundance. More MC4R mRNA was found in alpha MSH treated quails than the control group. The expression of AGRP and dopa decarboxylase was greater in the quails injected with the vehicle than the quails injected with alpha MSH. The mRNA from MC4R was greater in the alpha MSH treated birds. The quantities of neuropeptide Y and its substrates, corticotropin release factor, CRF, and its subtypes, POMC, ghrelin, mosatrin receptor, and MC3R and mRNAs were not affected by the treatments.
Discussion. Application of the finding that higher doses of alpha MSH caused the quail to eat less and drink less. Alpha MSH had same effects on food intake on the quail as rodents and broiler chicks. Alpha MSH caused an effect on the water intake of the quails that was not previously seen on any other animal subject. This suggests that quail have a lower threshold response in their sensitivity to anorexigenic effects of alpha MSH than broiler or layer chicks. Therefore, in future experiments, less MSH is needed for quail as opposed to the other birds. Quail could be used to further look at the appetite system on the chicks and rodents since they are very similar in food intake. Discussion. Application of the finding that CFOS immunoreactivity increased in the quails PVN and VMH when injected with alpha MSH. PVN and VMH activation appears to be conserved between quail and broiler chicks. This indicates that the effects of alpha MSH go through the PVN in both chickens and quails. Chicks showed an increase of CFOS immunoreactive cells in the DMN that was not seen in the quails, indicating some structure differences. Application of the finding that MC4R mRNA abundance decreased in the quails injected with alpha MSH. Some of the same genes were affected by alpha MSH injection in both the broiler chick study and the present one. This indicates that similar pathways are involved. Downregulation of AGRP mRNA may represent a response to the increase in anorexigenic melanocortin signaling. MC4R in both humans and quail mediate effects associated with weight loss. Therefore, mutations in MC4R in humans and quail can be associated with obesity. Critique. In order to make the article easier to understand, having less results to look for and discuss would make the implications of the data stronger. To make the experiment more accurate, more details could have been provided on the number of quail subjects used. The two sexes could also have been separated to see if any differences existed between them. The authors mentioned the lack of investigating the mRNA abundance in the VMH. This would be a good area of study for further experiments. Lastly, we wanted to comment on how neat the layout of the article was. It was helpful with understanding of the article. Conclusions. Finding that both the quails and the chicks had a lot in common as far as pathways when injected with alpha MSH could indicate the similarities between other birds. Since we have used birds in the past to look at human brain function, this could indicate similarities between humans and birds as well. By looking at food regulation, we could learn more about the mutations associated with obesity. These are our citations. Thanks for listening.